To examine engineered tissue constructs, traditional mechanical testing, which often results in the destruction of the sample, is not acceptable. This method uses microscopic magnetic resonance elastography, or micro-MRE, as a non-invasive technique for measuring the mechanical properties of small soft tissues. First, cells are seated onto a biomaterial scaffold to generate tissue. The tissue is suspended in agarose gel, and the tip of an actuator is placed into the gel. Then, to characterize the actuator, motion is transferred to the sample and detected using a laser Doppler vibrometer. The sample and actuator are transferred to a magnet, and magnetic resonance elastography images are acquired. Analysis of the resulting images shows the change in shear stiffness for both osteogenic and adipogenic constructs.
We first had the idea for this technique when we observed magnetic resonance elastography for the use of disease diagnosis and realized it could be extended to tissue engineering.
The main advantage of this technique over other existing techniques like mechanical testing is that it applies the non-invasive technology of MRI to measure mechanical properties of tissues.
The implications of this technique extend to tissue engineering because the knowledge of mechanical properties will ensure it is appropriate for its intended uses in bone and cartilage engineering.
Although these methods provide valuable insights into tissue engineering, it can also be used to diagnose diseases in different organs, such as liver fibrosis, traumatic brain injury, or brain cancer, for instance.
Visual demonstration of this